Hi guys, Alan here. So this is a continuation video from the one that we had on the introductory part, the cells, where we talked about the cell and we also looked at the cell theory and the guys who proposed the cell theory and the limitations that were experienced. So in this one we'll talk about the types of the cells, which are prokaryote and eukaryote, and then we'll also talk about the functions and organelles, and then we'll look at the differences. So let's get into it, guys. This is a summary video. So a prokaryotic cell is this one, and a eukaryotic cell is this one. Let's do this. So prokaryotic cells are usually between zero point 0.1 and 5 micrometers. So zero point 0.1 and 5 micrometers, they are small, and eukaryotes are generally larger because they are 10 to 100 micrometers. Prokaryote cells, they have higher surface area to volume ratio because they are smaller, which makes them able to obtain larger amount of nutrients through plasma membrane. Yeah. So the prokaryotes are smaller because they are zero point 0.1 to 5 micrometers. So this is the length that they have. And eukaryotes, they have a bigger length because it's 10 to 100 micrometers. Then let's look at the anatomy of a prokaryote. So this is a prokaryote cell, and in this case we have a bacteria cell anatomy. So a prokaryotic cell is a type of a cell that does not have a true nucleus or membrane bound organelles. So the example is the one that we have here, and the archaea is also an example of a prokaryote. So organisms that have prokaryotic cells, these are unicellular and called prokaryotes. So these prokaryotes, they're covered in cell membrane, which allows them to create a specific environment within the cytosol. So we'll talk about the functions in the future slide. So these are the things that we have here. We have the pairs, we have the cytoplasm, the nucleoid, the ribosome, and the flagella, the plasmid, plasma membrane, cell wall, the capsule. Okay, so this is a complex image. It's a bit detailed. So let's talk about the bacteria cells. So bacteria, these are single-celled microorganisms that are found near everywhere on Earth, and these are diverse in their shape because they are about 5 exponential 30, and bacteria, they are living in many different ecosystems on Earth, even in the bodies of humans in the gut, and they outnumber the human cells because the ratio is 1 to 10, which is a big difference. So let's now talk about the eukaryote cell. So this what we have here is a detailed structure of a eukaryotic cell, and in this case we have a animal cell. So we'll talk about the differences in the future slide, but in this case let's just look at the organelles that are there. So this is a detailed structure. We have the cytoplasm, we have the mitochondria, we have the nucleus, the lysosomes, the Golgi apparatus, peroxisomes, the centria. So it's a complex structure. It's very detailed, and we'll talk about these guys and the functions in the next slide. So let's talk about this. So this side we have organelles, and here we have functions. So the nucleus, so it contains genetic material for the cell, and ribosomes, these small complex proteins that assemble proteins from the mRNA, which is the messenger RNA. Then the next organelle is the Golgi apparatus. I don't know if you say Golgi, as long as you know the spelling, the pronunciation. So we define pronunciations and we made in spellings. So these, they package proteins into membrane bound vesicles for excretion from the cell. So these They are for excretion. So once they package proteins into the membrane-bound vesicle, then they're there for excretion as well. So the endoplasmic reticulum. So we have the SER and the RER. So they're different. So this one is rough, and the other one is smooth. So these they synthesize cellular materials, and they're divided into two. So the smooth and the rough endoplasmic reticulum. We have the nucleus. So the structures of the nucleolus. So the structures of the nucleolus that synthesize lysosomes, which is ribonucleic acid here. In this case, we have RNA. This RNA is single strand, and the structure of the nucleus that synthesizes ribosomes, which is the nucleus. Okay, so we have the mitochondrium, the powerhouse of the cell that performs cellular respiration. So it's the powerhouse of the cell. We have the vacuoles, the vesicles. So these are empty bags within the cell. The vesicles, the vacuoles, the empty bags within the cell. Then the vesicles, the packages of cell membrane that contain materials for the transport into or out of the cell. So packages of the cell membrane that contain materials for transport into and out of the cell. Cytoskeleton. The cytoskeleton. So scaffolding of tubes providing internal support for the cell. So they provide internal support. Cytoskeleton. Flagella, cilia, pili, they have the same function. Protein filaments that allow cells to move. Then these here, we have the cell walls, the chloroplast, the central vacuole in plants. So the cell wall, the cellulose structure supporting the cell. Then the chloroplast, photosynthesizing organelles for photosynthesis purposes. Central vacuole, a large water-filled sac in the middle of the plant cells. The ribosomes, so structures containing enzymes that break down unwanted materials. The centrosomes, organelles containing centrioles. Okay, so when we look at this, so the eukaryotic cell, okay, any organism that possesses a cell as a nuclear membrane that surrounds the nucleus, in which the chromosome, which are bodies containing of editing material are located. So also contain mitochondria, the powerhouse, Golgi apparatus, secretory devices, endoplasmic reticulum, and the rhizome, the digestive apparatus within many cells. So the rhizome, my interest is here. The rhizosomes, structures containing enzymes that break down unwanted materials. Okay, and then we look at this now, the final part of this video, the prokaryotic versus the eukaryotic. So prokaryotic cells and eukaryotic cells differ in a number of key features, including DNA, so composition and structure, organelles, types present and relative sizes, reproduction. Then this just involves more differs according to chromosome structure. Then the average size. So this talks about exception may exist. And then the way you can memorize this is using Dora the Explorer, the cartoon. So DNA, organelle, reproduction, average size. So DNA, then organelle, reproduction, and average size. So let's look at this now a bit. So this is what I would say. Dora, DNA, organelles, reproduction, and average size. So prokaryotic versus eukaryotic features. So prokaryotic On this side we have eukaryotics. So DNA is naked, and in eukaryotic, DNA bound to protein. So it's bound to a protein. On this side, it's naked. Then DNA is secular. DNA is linear. So remember we talked about the strands, and in this situation, in eukaryotics, they are what? Linear, straight, and here secular. Usually no night, usually no insurance, and here usually has insurance. And then organelles here, no nucleus, no membrane bound, 78 ribosomes as a nucleus, membrane bound, 80s ribosomes. So reproduction, binary fission, single chromosome, haploid, and then here it's diploid chromosome. So meiosis and mitosis. Then for average size, this is smaller. Excuse me. 1 to 5 micrometers, and these are larger because they are 10 to 100 micrometers. So this is all about this video, guys. Look up for more content, research for more. Thank you for watching this video, guys.